This conference will now be recorded.
Hello, good evening to all the students. Yes, good evening to all the students. Students, in last class, I think some students were present in the class. The topic was photosynthesis. In last classes, we discussed the topic that is photosynthesis. Photosynthesis, most students were in the class. How many students attend the class of the photosynthesis, the light reaction? How many students they attend the class of the photosynthesis? Yes, you attended the class. Okay. So in last class, students, we discussed the light reaction. I think सबको पता है. In last class, we discussed the light reaction. So today we are going to discuss the dark reaction of the photosynthesis. In last lecture, we discussed the light reaction. First of all, just take the revision. What is photosynthesis? Photosynthesis is the anabolic process in which the CO2 plus water molecules they combine together to form the glucose molecule and oxygen plus they release the energy. That is the reaction of photosynthesis. Just take the revision. First of all, just take the revision. After that, we will move on the next topic that is dark reaction. Okay, because maybe some students are new, so they are not aware with the topic and बहुत time पहले मैंने discuss किया था maybe इसीलिए. So today, first of all, just take the revision. What is photosynthesis? Anabolic process which takes place in the green plants, algae and some of the prokaryotes. Okay, the plants which contain the chlorophyll pigment, in them the process of photosynthesis takes place. Next is the site of photosynthesis. The next question that comes in our mind, that at which location the process of photosynthesis takes place. In the leaf, mesophyll cells are present. Okay, everyone knows that in the leaf mesophyll cells are present and they contain the chloroplast. And the chloroplast, what is chloroplast? Chloroplast is the autonomous organelle of the plant and it is double membrane. Outer membrane, inner membrane, okay? And in the chloroplast, stroma is present, thylakoid present, and grana are present. Thylakoid, grana, all of these are present in case of chloroplast. So in the green plants, mesophyll cells are present, they contain chloroplast. In the chloroplast, the chlorophyll pigment is present, and at this location, the process of photosynthesis takes place. Okay, so this is the structure of chloroplast. Outer membrane, inner membrane. This is the liquid part which is called stroma. Stroma is the site of dark reaction, and thylakoids, these are the site of light reaction. Group of thylakoid is called grana. Okay, the group of thylakoids is called the grana, and stroma is the liquid part. In the stroma, dark reaction takes place, but in the thylakoid, the process of light reaction takes place. Then students, we discussed that process of photosynthesis is divided into two reactions, light reaction and dark reaction, light dependent और light independent. Okay, सबको पता है कि जो process of photosynthesis होता है बच्चों अपना, it takes place in the two forms, light dependent as well as light independent. इसको हम light reaction भी बोलते हैं और इसको हम dark reaction भी बोलते हैं. First one is light reaction, other one is dark reaction. Okay, then light reaction is divided into two pathways, cyclic as well as non-cyclic. Okay, light reaction is divided into two pathways, cyclic as well as non-cyclic. So firstly we are going to discuss the साइक्लिक प्रोसेस। ये बच्चों हम सारा कर चुके, तभी इसका हम क्विक रिवीजन ले रहे हैं, फास्ट इसको कर रहे हैं ताकि हमारा फोटोसिंथेसिस का टॉपिक एटलीस्ट कंप्लीट हो जाए। ओके, ये लास्ट क्लासेस में हो चुका है, मोस्ट स्टूडेंट्स ने क्लास अटेंड की है, और जिसने अटेंड नहीं की है ये उनके लिए है, और जिनका हो चुका उनका जस्ट रिवीजन है ये। ओके, दिस इज द रिवीजन क्लास, बिकॉज बहुत सारे स्टूडेंट्स को ये टॉपिक मे बी।
Hello.
हेलो,
Sorry to interrupt you, ma'am. Here, all the three students, those who are there, they are from basically from South India, so they cannot understand Hindi, ma'am.
ओके ओके ओके, नो प्रॉब्लम सर। Students, light reaction, it is divided into cyclic as well as non-cyclic. The main one term, that is photoexcitation. We must know what is photoexcitation. Photo means light, excitation means molecules. When the light energy falls on the chlorophyll pigment, the antenna pigments, they absorb the light and electrons get excited, and these electrons are transferred from lower energy state to the higher energy state. This process is called the photoexcitation. Okay, what is photoexcitation? When the molecules, they absorb the light. Molecules means the reaction center of the pigments. Okay, reaction center, they contain the antenna pigments. On the antenna pigments, light energy falls, they absorb the energy, and electrons get excited from the lower state to the higher energy state, and this process is called photoexcitation. Then the next point that comes, photosystems. In light reaction, two photosystems are involved, photosystem 1 and photosystem 2. Okay, photosystems are involved, that is photosystem 2 and photosystem 1. Both of these photosystems, they contain the reaction center. Okay, you must know that in the light reaction, photosystems are present, and these photosystems contain the reaction center, and these reaction center absorb the light and they get excited. Okay, electrons get excited and they transfer from lower energy state to higher energy state. Okay, then in light reaction, two photosystems are involved, photosystem 1 and photosystem 2. Photosystem 1, it contains the reaction center P700. On the other hand, the photosystem 2, it contains the reaction center P680. Both the photosystems, photosystem 1 and photosystem 2, they contain the different reaction centers. Photosystem 1, it contains P700. Photosystem 2, it contains P680. Okay, I think this is clear to all the students. Then light dependent reaction. Firstly, we discuss the light dependent reaction. What is light dependent reaction? Light dependent reaction takes place in the thylakoid. In the chloroplast, the structure which is present is called thylakoid, and in the thylakoid membrane, the process of light reaction takes place. Okay, and what is the function of light reaction? It converts light energy into the chemical energy. Okay, light energy into the chemical energy. In the light reaction, ATP and NADPH are synthesized. In the light reaction, ATP and NADPH are synthesized. Clear? What is the main difference between light reaction and dark reaction? In the light reaction, ATP and NADPH are synthesized, and these ATP and NADPH are used in the dark reaction. So this is the main difference. Okay, and light reaction always takes place in the presence of light. On the other hand, dark reaction, it may take place in the presence of light or maybe in the absence of light. Okay, for dark reaction, light is not required at all the time. Light is not required, and dark reaction doesn't mean that it takes place in the dark. Okay, it may takes place in light or in dark. In any of the condition, dark reaction can take place. But light dependent reaction, that is light reaction, it always takes place in the presence of light, and it occurs in the thylakoid. Thylakoid, we see the structure of chloroplast, and in the chloroplast, the thylakoid was explained. So, light reaction occurs in the thylakoid. It converts light energy into the chemical energy, and ATPs and NADPH are synthesized. Then, next point, that is Light reaction is divided into non-cyclic and cyclic photophosphorylation. First of all, let's discuss the non-cyclic photophosphorylation. In non-cyclic photophosphorylation, what happens? See the diagram. Students, I think you are able to see the diagram. Let's discuss the diagram. Okay. This is the pathway of non-cyclic photophosphorylation. This pathway is also called as Z scheme. Z scheme because the molecules are moving in the form of Z. Okay. So this pathway is also called as Z scheme and it is non-cyclic photophosphorylation. Then what is the process of non-cyclic photophosphorylation? In non-cyclic photophosphorylation, photosystem 2 and photosystem 1 both are involved. Okay, photosystem 2 and photosystem 1 both are involved. First of all, what happened? Let's see the diagram. From here, we discuss the diagram. It is the photosystem 2. The photosystem 2 has the reaction center that is P680. Light energy fall on this P680. Okay, electrons get excited and these electrons are accepted by the primary acceptor. From the primary acceptors, electrons are transferred to plastoquinone. From plastoquinone, transferred to the cytochrome complex, then plastocyanine and P700. Clear? First of all, light energy falls on photosystem 2. Electrons get excited, transfer to the primary acceptor, from primary acceptor, plastoquinone, from plastoquinone to the cytochrome complex, from cytochrome transfer to the plastocyanine, then P700, that is photosystem 1 and reaction center is P700. Again the light energy falls on the system, electrons get excited, accepted by the primary acceptor, transfer to the ferroxin and NADP reduced into the NADPH. So, in light reaction, in non-cyclic photophosphorylation, NADPH and ATP both are formed. Then the next question that comes in our mind, the photosystem 2, from where it gets its electrons. Photosystem 2, it takes its electrons from the splitting of water. The water molecules get break down and it releases 2 electrons, oxygen plus hydrogen. These electrons are accepted by the P680. And light energy falls on this P680 and these electrons get excited. Okay, the question that comes in our mind, the P680, the reaction center of photosystem 2, from where it is getting the electrons. Yes, students, the question आता है अपने mind में, the question comes in our mind, from where the P680 is getting the electrons. P680 is getting electrons from the hydrolysis of water. Water molecules get split here and they release 2 electrons, oxygen plus hydrogen. So electrons are donated or given by splitting of water or hydrolysis of water. I think this is clear to all the students. Message me if any one of you have any doubt, you can message me. After that we will move on the next process. Clear to all the students? Clear. Okay. Okay, clear to all the students. Let's move on the next process, that is cyclic photophosphorylation. What is cyclic photophosphorylation, students? From the name, it is clear what is cyclic. Electrons move in the form of the cycle. Okay. In cyclic photophosphorylation, only one photosystem is involved. That is photosystem 1. Okay. Only one photosystem is involved. In non-cyclic photophosphorylation, photosystem 1 and photosystem 2 both are involved. But in case of cyclic photophosphorylation, photosystem 1 is involved. Only photosystem 1 is involved. Okay. Light energy falls on this photosystem 1. Electrons get excited, accepted by the primary acceptor. Then ferroxin, cytochrome complex, plastocyanine, and back to the photosystem 1. This is the process of cyclic photophosphorylation. Electrons are moving in the form of the cycle. Electrons released from here and come back on this point. So it occurs in the form of cycle. Cycle is going on. So this process is called cyclic photophosphorylation. Okay. Then next point. That was a revision class. In last class we discussed these points, but today we are going to discuss the new topic, that is light independent reaction. The new topic, that is light independent reaction. Okay students, I think light reaction is clear to all the students. Now move on the light independent reaction, or it is also called dark reaction. Dark reaction or light independent reaction. First point, main point, it is called Calvin cycle. Why it is called Calvin cycle? Because it was discovered by the scientist named Melvin Calvin. So this cycle is called the Calvin cycle. Other name of this cycle is C3 cycle. Students, other name is C3 cycle. C3 cycle. Why it is called C3 cycle? Because first stable product that is formed is the first stable product that is formed is 3-carbon compound, that is phosphoglycerate. First molecule that is formed in this reaction is 3-carbon molecule, which is a phosphoglycerate, PGA. Okay. So this reaction is called C3 cycle, as well as dark reaction, as well as Calvin pathway. Don't get confused students, what is dark reaction, Calvin cycle, C3 pathway? All of these are the same. This cycle is called C3 cycle and it takes place in the stroma of the chloroplast. Light reaction takes place, bacho? Just answer me, light reaction. Light reaction takes place, the location of light reaction, site of light reaction. Answer me. Thylakoid, yes, very good. Vidushi, very good. Light reaction takes place in the thylakoid, but dark reaction, it takes place in the stroma. Okay. In this process, no light energy is required. Light energy is not required. Okay. As in case of light reaction, we see always occurs in the presence of light. But in dark reaction, it is not the rule. Dark reaction doesn't occur in the presence of light. It may take place in the presence of light, or it may take place in the absence of light. Okay. In dark reaction, ATP and NADPH. ATP and NADPH, which are produced by light reaction, are used. And this reaction needs the enzyme Rubisco enzyme. This reaction, this pathway required the enzyme, that is Rubisco enzyme. So just discuss some points, some important points about the Rubisco. Rubisco is the most abundant enzyme in the plant world. Okay. Rubisco is the most abundant enzyme. Why it is most abundant enzyme? Any one of you can tell me? What is the reason that Rubisco enzyme is the most abundant enzyme? Full form of Rubisco is ribulose bisphosphate carboxylase oxygenase. Okay, ribulose bisphosphate carboxylase oxygenase. Rubisco has both the properties of carboxylase as well as oxygenase. Means it can bind with CO2 as well as it can bind with oxygen. Okay, that's why Rubisco is the most abundant enzyme, because it has the capability to bind with oxygen as well as CO2. Full form of Rubisco, ribulose bisphosphate carboxylase oxygenase, means it can bind to CO2 as well as oxygen. That's why Rubisco is the most abundant enzyme in the plant world. Okay, so these are the points of light reaction. Light independent reaction or dark reaction. Calvin cycle, also called C3 cycle, okay, occurs in the stroma. It doesn't require any light. The ATP and NADPH, which are produced by the light reaction, are used in this pathway. In this cycle, all these ATP and NADPH are used. It occurs in the presence of enzyme called Rubisco. Okay, occurs in the presence of enzyme Rubisco. What is Rubisco? Ribulose, bisphosphate, carboxylase and oxygenase. Let's discuss the cycle. Just a minute. First of all, see the diagram. This is the complete cycle of C3 pathway. In C3 cycle or dark reaction, mainly three processes takes place. That is carboxylation, reduction and regeneration. These three processes takes place in the process of dark reaction, carboxylation, reduction and regeneration. Okay. First step called carboxylation. In the first step, 6 CO2 molecules or 3 CO2 molecules, because this reaction takes place 6 times to form the 1 glucose molecule. So here 3 CO2 molecule plus 3 RUBP. 3 CO2 plus 3 RUBP molecules, they combine together to form the 6 molecules of PGA. 3 CO2, 3 RUBP, both are combined together and they form the 6 molecules of PGA. It occurs in the presence of enzyme Rubisco. This step is called carboxylation step. Come on the next, let's come, yes. First step, phase 1, that is carbon fixation, or it is also called as carboxylation. Okay students, in first step, CO2 molecule combine with RUBP, ribulose 1,5-bisphosphate, and they form the short 6 molecule intermediate, that is PGA. Okay, PGA molecules are formed, that is phosphoglyceraldehyde, that is 3 carbon compound. Next process is these 3 PGA molecules get converted into the 1,3-bisphosphoglycerate. Okay, by using 6 molecules of ATP. ATP molecules are converted into the ADP molecules. Then this 1,3-bisphosphoglycerate reduced into the glyceraldehyde 3-phosphate by using 6 NADPH. So students, 6 ATP and 6 NADPH molecules are used up to this step. The second step is reduction. What is reduction? Carboxylation, I think clear to all the students. In carboxylation, CO2 and RUBP, they form the PGA molecule. Okay, that is 3-phosphoglycerate, PGA molecule. And this PGA molecule reduced into the glyceraldehyde. This process is called reduction step because phosphoglycerate molecule is converted into the aldehyde molecule. So this is called the reduction process. Okay, because aldehyde molecule, phosphoglycerate molecule is reduced into the aldehyde molecule, glyceraldehyde. So in this process of reduction, 6 ATP and 6 NADPH molecules are used. This is the second phase. Reduction phase. In reduction phase, this process takes place. Up to here, this is the second phase, phosphoglycerate to phosphoglyceraldehyde. So the glycerate molecule get reduced into the aldehyde molecule. This process is called reduction process. And in this process, 6 ATP and NADPH molecules are used. Clear students? 2 steps are clear? Carboxylation, reduction. These 2 steps clear to all the students? In C3 cycle, mainly 3 steps are present. Okay, clear. If any one of you have any doubt, then you can ask. Okay. So, in process of C3 cycle, mainly 3 steps are present. First is carboxylation, second is reduction, and third is regeneration. We discussed 2 steps, that is carboxylation. Carboxylation, from the name it is clear, CO2 molecule combined with RUBP and they form the phosphoglycerate molecule. And in the reduction process, this phosphoglycerate molecule reduced into the aldehyde molecule, that is phosphoglyceraldehyde. Then third process is regeneration. Students, third process is regeneration. At this point, we discuss the regeneration. What is regeneration process? In the regeneration process, this glyceraldehyde molecule, out of these 6 glyceraldehyde molecule, 1 molecule is used for the formation of glucose. Okay? And 5 molecules, they regenerate the RUBP molecule. And in this process, again 3 ATPs are used. Clear? 3 molecules RUBP, 3 molecules CO2 form 6 PGA. These 6 PGA molecules, they get converted into the 6 glyceraldehyde molecule. Out of these 6 glyceraldehyde molecule, 1 molecule get converted into the glucose and 5 molecules. Out of these 6, 1 form the glucose and the remaining 5 molecules, they regenerate the RUBP. And in this process, 3 ATPs are used. So total, how many ATPs are used? 6 ATPs at this step and 3 ATPs at this step. Total 9 ATP and 6 NADPH. Okay. And this cycle takes place in the, here 3 CO2 comes and 3 RUBP comes. But carbon molecule is of 6 carbon. So we have to complete this cycle in 2 turns. In first turn, 9 ATP and 6 NADPH are formed. In the second turn, we multiply the full output with 2 more. 18 ATP and 12 NADPH molecules are formed. The end output of the last output of the dark reaction is 12 NADPH molecule and 18 ATP molecules. Because students, 3 CO2 and 3 RUBP, but we have to complete, because carbon is, glucose molecule is C6, 6 carbon molecules are required. Here only 3 molecules get entered in the cycle. So we have to complete this cycle in 2 turns. In 2 turns, we form total 18 ATPs and 12 NADPH. Okay, I think clear to all the students. C3 cycle, it is called Kelvin cycle or dark reaction or C3 cycle. Okay, light independent Kelvin and C3 cycle occurs in the presence of enzyme Rubisco. Then Rubisco is the most abundant enzyme in the plant world. Why it is most abundant? Because it has both activities, carboxylase and oxygenase. It has both the activities. Okay, then C3 cycle occurs in 3 phases: carboxylation phase, reduction phase and regeneration phase. In carboxylation, CO2 molecule is formed. In case of reduction, aldehyde molecule, phosphoglycerate molecule is reduced into the aldehyde molecule. Okay, and in regeneration molecule, from the glyceraldehyde molecule, glucose and RUBP, this RUBP molecule get regenerated. So this is the process of dark reaction or C3 cycle. Clear? Clear? Okay. So next, move on the next cycle, that is C4 cycle. Okay. Next cycle is C4 cycle. Then what are the limitations of C3 plant? C3 plant की limitations क्या होते हैं? What are the limitations of C3 plant? In hot condition, they are not able to grow. In hot and arid condition, the stomata get closed and they prevent the loss of water. Okay. And RUBP molecule, it can combine with oxygen as well as CO2 molecule. That is the lossful process. We will discuss this point in the further next slide. The next pathway is C4 pathway. What is C4 pathway? C4 pathway, it is also called H and Slack pathway. It is also called H and Slack pathway because it was discovered by H and Slack scientists. In this process, C4 process, the first stable product is 4-carbon compound. Okay, that is oxaloacetic acid. In case of C4 plants, the first stable product is 4-carbon compound, that is oxaloacetic acid. The features of C4 plants. What are the main features of the C4 plant? They possess the crane's anatomy. Okay, students, in case of C4 plants, crane's anatomy is present. Then next question comes in our mind, what is crane's anatomy? Crane's anatomy means mesophyll cells and bundle sheath cells are present. The bundle sheath cells, they are always surrounded by the mesophyll cells. Okay, in the center, bundle sheath cells are present, and these bundle sheath cells are surrounded by the mesophyll cell. This anatomy is called crane's anatomy. Okay, the anatomy is called crane's anatomy. It is students. Sorry for inconvenience. Okay, the crane's anatomy is one of the most important feature. Okay, crane's anatomy is one of the most important feature of the C4 plants. Crane's anatomy means the bundle sheath cells are surrounded by the mesophyll cell. Then we will discuss in next slides why this process takes place. Okay, this is the crane's anatomy. These are the bundle sheath cell, and around these bundle sheath cell, mesophyll cells are present. These are the mesophyll cells, and they surround the bundle sheath. Then next feature about C4 plant, they grow in the tropical, subtropical regions. Okay, they grow in tropical as well as in the subtropical regions. They are able to grow at high temperature. The C3 plants, they have the limitation. What was the limitation? At high temperature, their stomata get closed to prevent the loss of water. But in case of C4 plant, this limitation is not present. Why? Because in C4 plants, crane's anatomy is present. Okay, in C4 plants, the crane's anatomy is present. That is the main reason why in case of C4 plant, this water loss doesn't take place, and this process also takes place in the high temperature condition. High temperature condition. Okay. So this is the process. These are the mesophyll cell and these are the bundle sheath cell. Keep in mind that in the C4 pathway occurs in two locations, mesophyll cell as well as in bundle sheath cell. On the other hand, C3 pathway always occurs in the mesophyll cell. Okay, this point keep in mind that C3 pathway occurs in mesophyll cell, but C4 pathway, it occurs in mesophyll as well as in bundle sheath cell. This anatomy is always called the crane's anatomy. What is the process of C4 pathway? In C4 pathway, first of all, CO2 molecules, they combine with PEP, phosphoenol pyruvate, in the presence of enzyme PEP carboxylase. It takes place in the presence of enzyme PEP carboxylase. Okay, and they form the OAA, oxaloacetic acid. This oxaloacetic acid converted into the malic acid, aspartic acid, and these malic acid and aspartic acid transport into the bundle sheath cell with the help of plasmodesmata. Okay, students, I think you are getting my points. What is the process? In case of C3 cycle, CO2 combines with RUBP. But in case of C4 pathway, CO2 combines with phosphoenol pyruvate, PEP. It combines with PEP. In the presence of enzyme PEP carboxylase, it forms OAA, that is oxaloacetic acid, which is 4-carbon compound molecule. So it is called C4 pathway. Why it is called C4 pathway? Because the first product is 4-carbon molecule. Okay, OAA is formed, oxaloacetic acid. This OAA get converted into the malic acid or aspartic acid. This malic acid or aspartic acid get transported into the bundle sheath cell, and in the bundle sheath cell this malic acid get converted into the pyruvic acid and CO2 is released, and in the bundle sheath cell Rubisco enzyme is present. So this CO2 molecule combines with the RUBP and the process of Calvin cycle takes place. So in the mesophyll cell, PEP carboxylase enzyme present. In the bundle sheath cell, Rubisco enzyme is present. So in the presence of Rubisco enzyme, Calvin cycle takes place in the bundle sheath cell. This malic acid converted into the pyruvic acid. This pyruvic acid again transported into the mesophyll cell, and this process again takes place. So this is the cycle of the C4 plants. This is the C4 cycle. Notes eleutheria means what is notes eleutheria? What is this, students? I am not getting your point. BSL Shalini. Notes eleutheria. I am not getting your points. What you want to say? Okay. Clear? C4 pathway occurs in mesophyll cell. Again, listen, students, very carefully. C4 pathway occurs in mesophyll as well as in bundle sheath cell. In the mesophyll cell, PEP carboxylase enzyme is present, and in the bundle sheath cell, Rubisco enzyme is present. Okay, so PEP carboxylase enzyme, in the presence of PEP carboxylase enzyme, CO2 molecules they combine with PEP, phosphoenol pyruvate, and form the oxaloacetic acid. This oxaloacetic acid forms malic acid as well as aspartic acid. This malic acid or aspartic acid, they get transferred into the bundle sheath cell with the help of plasmodesmata. This malic acid converted into the pyruvic acid, okay, and CO2 is released. The CO2 present, Rubisco is present. So Rubisco enzyme catch the CO2 molecules and Calvin cycle takes place. So in bundle sheath, Calvin cycle occurs, and in the mesophyll cell, formation of malic acid takes place. So this is the process of C4 pathway. Then we discuss the one term, that is photorespiration. What is photorespiration? Students, photorespiration occurs in C3 plant. Photorespiration occurs in C3 plant. Why it occurs in C3 plant? Because in C3 plant, Rubisco enzyme is present. And this Rubisco enzyme has both the properties of carboxylase as well as oxygenase. If this Rubisco enzyme binds with oxygen molecule, if the concentration of oxygen is very high, then it may bind with the oxygen molecule, not with CO2. Clear? What I want to say, in photorespiration, this Rubisco, because Rubisco enzyme has both the properties of binding with CO2 as binding with oxygen. In any condition, when the concentration of CO2 is very less and concentration of oxygen is very high, at that time, this Rubisco enzyme, it get binds with the oxygen molecule. And when Rubisco get binds with oxygen instead of CO2, at that time, it becomes the wasteful process because phosphoglycolate molecule get formed. Okay, phosphoglycolate molecule get formed, no glucose molecule will form, no ATPs are used, no NADPH are used, nothing get used because it is the wasteful process. So photorespiration becomes the wasteful process. But in case of C4 plant, the process of photorespiration doesn't take place. Why? Because in C4 plant, Crane's anatomy is present, and in bundle sheath cell, Rubisco enzyme is present. And in the bundle sheet, CO2 concentration is always very much high. Why CO2 concentration is very much high? Because malic acid is converted into pyruvic acid at the time CO2 is released. So Rubisco enzyme doesn't have any chance to bind with oxygen. उसके पास कोई option नहीं होता. It always binds with CO2. When Rubisco binds with CO2, then the process takes place in the proper way. Glucose is also synthesized. But in case of C3 plant, C3 plant में students क्या था? In case of C3 plant, trans atom is not present. Yes, see, this is the C3 pathway. In this Rubisco enzyme, it may either bind with CO2 or it may bind with oxygen. When it binds with oxygen, the process of photorespiration takes place, and when it binds with CO2, then the normal process takes place. But in case of C4 plants, this process doesn't take place. Why? Because Rubisco enzyme is present in the bundle sheath cell. And in the bundle sheath cell, concentration of CO2 is very much high. Okay? Concentration of CO2 is very much high. So there is no chance of binding Rubisco with oxygen. It always binds with CO2. So, photorespiration. Photorespiration occurs when the CO2 levels is very low. It occurs in the hot and dry conditions. On hot and dry days, stomata prevent excess of water, fixed CO2, oxygen ratio is very much high. At that time, the Rubisco enzyme binds with oxygen. Rubisco combined with oxygen instead of CO2, due to which phosphoglycolate is produced. Toxic molecule. Phosphoglycolate is the toxic molecule which get formed, due to which no glucose molecule is synthesized. As a result, the process of photorespiration is wasteful process. Clear? Students, clear? C4 pathway and C3 pathway clear to all the students, and photorespiration. It is the additional term. If any one of you have any doubt, you can ask. In C3, C4 and photorespiration, now it's your turn. 10 minutes. If you have any doubt, you can unmute your mic and you can ask the doubt. Students, 5 minutes, 10 minutes are given to you if you have any doubt. Unmute your mic and you are free to ask any question. C3 and C4 pathway and photorespiration. Photorespiration is also called C2 pathway. Okay. Sometimes they ask about the C2 pathway and location of photorespiration, chloroplast, mitochondria and peroxisomes. It takes place in chloroplast, mitochondria and peroxisome. Today, mainly C3 pathway and C4 pathway. These are the main pathways. C3 pathway, 3 carbon molecule PGA get formed. In case of C4 pathway, 4 carbon molecule OAA get formed. Okay, and this OAA converted into the malate and malic acid, transported to the bundle sheath cell. In bundle sheath cell, Calvin cycle takes place. So in case of C4 cycle, Calvin cycle also occurs in the bundle sheath cell. Why? Because in the bundle sheath cell, RUBP enzyme is present. Okay, students, thank you. Sir, from my side, I have done the topic.
Okay, ma'am. Okay, ma'am. Students, if you have any doubts regarding today's topic, whatever being taught today, just ask your doubts. If you don't have any doubts, you can just comment yes in the comment section. Just let us know. Okay, thank you for responding. So that's all for today. I hope you all enjoyed, and you can slowly leave the meeting. You will get the video link and PPT today's. So thank you, Pooja ma'am. It was really informative.